Okay, good afternoon, everyone. This is our lecture 12. Today, we're going to continue our discussion on dimension reduction, which is a big category of methods for unsupervised learning. So for unsupervised learning, our input is always only a data matrix or feature matrix, where you have rows as n data points, or in statistics, we call them n observations. For the columns, we correspond them to np features. So it's an n by p matrix. So whenever we write the rows out, we use the lowercase x1 transpose to xn transpose. Each xi transpose is the ith row. When we write down the columns, we use the bigger x1 to xp. So that's for a feature vector. Each column here contains the n observations. So the general goal of doing dimension reduction is to find a way to embed those n data points into a lower dimensional space. Instead of a p-dimensional space, we want to find a much smaller k so that we can represent these n data points by an n by k matrix. Okay, so just as a quick reminder, we know that an important property about linear algebra is that for each row, you can always encode it as a point in an n-dimensional Euclidean space, right? So each in the row, the n values are the n coordinates in the n dimensions. You can also consider that point to be a vector. So the vector way of thinking about an n-dimensional vector is equivalent to the point representation. So what's the vector? The vector is that in that n-dimensional space, you draw a vector from the origin to that point. That direction is the vector. So these two ways are exchangeable. So each point corresponds to a unique vector. And the vector representation is very important because it gave us that natural representation or geometric representation of inner product. So whenever we have two n-dimensional vectors, we do their inner product or dot product. That means we multiply their two values in each dimension, and we take the sum for those products, the n product. That's what we call the inner product. And that inner product has the geometric interpretation as the cosine of the angle between the two vectors. So if the two vectors are called orthogonal, their inner product is equal to 0. So if you know that geometric representation, it will be easier. So for here, we can think of the original data points in a p-dimensional space, and we want to find a k-dimensional space so that we can project these endpoints to the k-dimensional space. And how does the projection work? So to find the projection, we basically need to know how each basis in the new space from 1 to k corresponds to the original p basis. So we write each ki as a linear combination of x1 to xp. And for these weights, you see, I can call each one as the phi 1, which contains the weights of z1, to phi k, which contains the weights of zk. And each phi is called a loading vector, and correspondingly, each z is called a score vector. So that's the coordinates of the endpoints in each new dimension. So in our last lecture, we spent quite some time to say, how do we find the first PC? The goal of PCA is to find Z1 so that it has the largest variance. Okay, so we want to find phi 11 to phi p1 to maximize the variance of Z1. So that is the variance of Z11 to Zn1. When we assume without loss of generality that the average is 0 across data points in each original dimension. Okay, so if we have every column of X to be centered at 0, then we know the Zi, these n values are also centered at 0. So therefore, we can say that to maximize the variance in Z1 is equivalent to maximize Z1 inner product with itself. So therefore, we can write this continue and continue. And then we find that the problem by the Lagrange multiplier approach can be written as an eigen decomposition problem for X transpose X, the P by P matrix, the feature by feature matrix. So then we said, okay, this eigen decomposition problem will tell us the maximum variance in PC1 is actually the largest eigenvalue of X transpose X. So to find that eigenvalue and its corresponding eigenvector is our first loading matrix. So we just do eigen decomposition and we get a result. So in the end, what we said in our last lecture is this relationship between eigen decomposition and singular value decomposition. So when we say eigen decomposition, we talk about a symmetric matrix. Here is X transpose X, the P by P feature by feature matrix. But when we talk about singular value decomposition, this can be applied to any matrix of any dimension. So we can do that on the original matrix, feature and the data matrix. Then in this case, the right singular vectors will be our loading vectors. And our variances or the eigenvalues of X transpose X happens to be the squares of the singular values of X. So if these are something new to you, probably you can just put them aside for now. The key thing to know is that PCA is a procedure that can do the variance decomposition. If you take the variance in each PC by adding them up, the more PCs you have, the more variance you can explain in your original data. So remember, when we talk about variance, it's always about how these n data points are spread around their center. So, okay, that's what we call about variance. So when we set k to be equal to p, in other words, after we do the transformation, we still keep the same number of dimensions as in our original data. So we don't do any dimension reduction. Then we keep all the variance. Then the percent of variance explained, short as PVE, will be equal to 1. But if we only keep a small number of PCs, we will have a small PVE. So how many pieces to keep? This is a very challenging question. It's usually based on this elbow plot. We plot how much PVE can be explained by how many pieces in total, and then we decide whether that's a reasonable K we want to keep. So that's the question. And if you check the SURAT tutorial, which you will probably use for your final project, there is a PCA dimension reduction for single cell data. So for single cell data, we usually say keep K equals 30, but the original P might be 20,000. That's the number of genes. So when you do the PCA, the goal is to remove unwanted variation that is probably not of biological interest, and we only keep the major components to explain the variance, so that hopefully our data later analysis will only be based on the K equals 30 dimensions. And that will make a lot of analysis much easier, because it's usually much more difficult to deal with high dimensional data like P equals 20,000. So a question I got after last lecture is about the interpretation of loading matrix and the score matrix. So let's continue there. So given my notation, so we have X as M by P, right? So we know we want the Z, which is M by K. So, you know, by matrix multiplication, Z must be multiplied by something as K by P. So I can just call that probably using my own notation. Okay, so this is a K by P matrix. Then what is W exactly? You know, it has K rows. And here the K rows are actually what we have as the loading. So we'll have phi 1 transpose to phi k transpose. If you don't recall what phi is, here they are. So phi 1 contains the weights of x1 to xp for the new dimension 1. Phi k contains the weights for x1 to xp for the new dimension k. So I'm putting them horizontally to get this W matrix. So in other words, each row is a loading. So I can say this is loading 1 to loading vector, loading K. Okay, and then how do we interpret this result? In some textbooks or papers, people may write their transposes. So if you write X as P by M, then on the right-hand side, you will have a W transpose as P by K matrix times Z transpose that is K by M. So that's also possible. Just make sure that you are clear about the dimensions. So here, how do we interpret this? So each loading vector is the contribution of the original P dimensions, right? We have P here and we have K here. Original P dimensions to each new dimension. So we can expect that for the original dimension, let me use the concrete example, genes. If some of your genes are highly correlated, then they will make large contributions to one dimension in the loading matrix. So you can think of an extreme case, that is, if one gene's expression is always 2 times the expression of another gene, which means that they're perfectly collinear or perfectly correlated. So in that case, using one gene versus using 2 genes does not give you any difference in terms of information. So in that extreme case, we should combine these 2 genes into one new dimension. One new dimension is enough. So that's why we are going to see they have non-zero loadings in one row here. Okay, so please note that PCA, the loadings could be positive or negative, but we care about the magnitude because the magnitude reflects how much information do you use from that original basis. So some people will look at the loading matrix to try to explain what the meaning of each new dimension is. Because you know that here, when you write a Z, each data point, let's use the analogy in single cell, each cell here will have encoding in K new dimensions. So from a biologist perspective, you want to know the meaning of each new dimension. So then what people usually do is that they will plot each new dimension against an original dimension. So by looking at the loading matrix, you can also see that information. So if this loading is really having a large value for, say, if this is the new dimension K, new dimension 3, if you see the original dimension 3, that means gene 3 has a large loading in this new dimension, then you can say, oh, my new dimension 3 is dominated, or my PC3 is dominated by the gene 3. But sometimes maybe the explanation does not come from the original P genes. So for example, in single cell data again, so when we do the dimension reduction, we obtain each new dimension. And for the cells, we can plot, let's say, we take the column 1 of C1, of Z1. So let me write this out. So I can write Z as Z1 to ZK, the K columns. So if I plot, make a plot of this, I plot Z1 against a feature that I don't include in my X. Let me say cell size, cell library size. That is the number of reads in each cell. So I will have dots here. So each dot represents one cell. So what I'm doing is that I'm plotting the score of the cell in Z1 and the cell library size. So if I see a very strong correlation here, then I would say, oh, my first PC is dominated by the cell library size. Then depending on whether I want to keep this information or not, I would do my analysis differently. So if you think this information is something not of your interest, that is, you don't care about the differences among cells in library size, then in your downstream analysis, you could just treat this Z1 as something to be removed. So you could just discard Z1 and continue from start from Z2 to ZK. Use the remainder of the Z matrix, just a K minus 1 columns for your analysis. So you don't use Z1 at all. Or some people might want to say, this Z1 seems to be a confounder. I want to remove from my data. So then what you can do is that you can take each column in X, X1 to XP, regress that Xj, each J on Z1, so that you're just taking the residuals as your new data. So if you want to remove Z1 from X, okay? So here X is going to be written out as X1 to XP. What you could do is that you would do Xj, do the regression on Z1. Treat Z1 as the thing to be regressed out, and then you will get residuals. You get residuals, let me call that as Rj. So here I can do that for every J, for J go from 1 to P. Then your new data to be used for analysis could be just R1 to RP. So you could do that. That's also something common in bioinformatics. If you want to remove the effect of something, then you can do linear regression on that to remove the linear effect. Okay, so I want to say that although we can examine the loadings for interpreting each new dimension, but this is not really easy for PCA, because if you know you haven't done PCA before, look at the loading matrix, then each row will contain a lot of non-zero entries, and some entries are non-zero but quite small. So in that case, it's not very straightforward how to interpret the results exactly. So motivated by this, there is a variant proposed for PCA that is called NMF. So that's the thing I'm going to introduce next. It's a little bit different from the order in our notes, but I feel like talking about NMF after PCA is better. Oh, and by the way, I forgot to say an important thing. That is, given this matrix W, this matrix W, okay, is a P by K matrix. It can be used as a projection matrix. So this can be a projection matrix. What does that mean? It means that if I have new data, given new data, measure for the same P features but different rows, I call it X prime or X star. Yeah, don't confuse the prime with transpose. So if this is an N star by P matrix, okay? So given this, and the P correspond to the original P, so has the same columns as X. Okay, if this is your new data, you want to project it to the same k-dimensional space. So you can compare these N star cells with the original N cells. Then you can take the W you obtain by performing PCA on X. Then you can have Z star equals X star times W. You see, this is N star by P. This is P by K. Oh, this is W transpose times K by P. Yeah. So you times P by K. So W transpose is P by K. So multiplying this together, you are going to have Z star as N star by K. So you can always do this. And also a nice property about W, that's the property about the rows of W are eigenvectors. So they're orthogonal to each other. So what you will have is when you do W times W transpose, okay? So in other words, you are going to have this phi 1 transpose to phi k transpose times W transpose will give me phi 1 to phi k. Okay, so this will give you what? This will actually give you a diagonal matrix with ones in the diagonal and 0 elsewhere. So this is a k by k matrix, or we can write as an identity matrix of k dimensions. The reason is that when I do the multiplication, I'm going to do phi 1 transpose times phi 1. That gives me the first entry. Phi 1 transpose times phi 2 give me the second entry, and so on. So because of the orthogonality, I am only to have 1 when phi 1 transpose is multiplied to phi 1, or phi I transpose is multiplied to phi I. For other things, I'm going to have 0. So this is a very important property about W as a projection matrix. So you can consider it as what? So whenever we multiply a matrix by doing the right multiplication like this. So essentially we are transforming the data to, we are transforming the data from P dimension to K dimensions, okay? So we're doing that. And whenever we are actually projecting the, whenever we do the left multiplication, so we are projecting the, let me see. So whenever we do the right multiplication, yeah, we keep the rows. I should say this. We keep the rows, but we just have the rows with new columns. That's what we mean by right multiplication. But for left multiplication is that we keep the columns, but we change the rows to different rows. So that's the intuition. And then here the W times W transpose, this is what? So this is from, okay, P by K is here. This is K by P here. So this means what? So it means that whenever I multiply a matrix on the right, probably I use a different color. Okay, so let me think about a case. Okay, so let me call this Z transpose, okay? Because Z transpose, I know it's K by M, then I have P by K, K by P. So this will give me the same Z transpose. So that means what? It means that for this matrix, which encodes, here the columns are my n data points, the rows are the new K dimensions. When I do the left multiplication, I keep the n columns, but I change the rows to be P rows in the P space, P dimensional space. And then I do the multiplication again by going from the P-dimensional space back to the K-dimensional space. So what this says is that when I do this from low dimension to high dimension, and then from high dimension to low dimension, I don't lose any information. So this is actually invertible, okay? So I can still go back. This is an important property about W. So geometrically, it tells you that whenever you are in these K dimensions, going to a higher dimension, you can go back to a low dimension. Okay, but as I said, the loading matrices here, the loading vectors are not sparse. So the issue with PCA is phi 1 to phi k are not sparse, which means that they do not have entries that are exactly zeros, but you will have many small entries, like small positive, small negative entries. So that makes the interpretation not very good. Interpretation not very easy. But it indeed does a very good job for keeping the variance. So if you are interested in using the Z matrix after the projection, yeah, PC is very good. It retains a lot of the information, the major information in your data variance. But if your goal is to interpret the loading vectors, then PCA does not give you a clear observation or clear interpretation unless you do some truncation on those loading vectors. That means if the absolute value is not greater than something, I truncate it to 0. So we know that's not very ideal because that depends on how we set the truncation threshold. So motivated by this, in 1999, there was a Nature paper proposed an algorithm, which is still popular nowadays. This is called non-negative matrix factorization, short as the NMF. So as the name speaks, we know it has non-negativity constraint. Okay, so what it aims to do is that it still aims to find the same decomposition into a lower dimension. So still have M by P. We want to find Z times W, which is M by K and K by P. But what we require is that, or want this to be approximate, what we require is that both Z and W must be non-negative in every entry. So we need this. Probably I need to write it out more clearly, like Z, I, K, and W, K, J must be non-negative for all the I go from 1 to M, and K go from 1 to K, and J go from 1 to P. So we want all the entries to be non-negative. That's the constraint. So therefore, the optimization problem becomes trying to find the minimum of this this loss function. So when we write x minus z times w, and this norm is a matrix norm. We call this Frobenius norm. So what this means is that if I have a matrix X, it's F, it's sometimes called F norm. It's basically the summation I from 1 to N, J from 1 to P, Xij squared. I take the square of the entries, and then I take the sum for rows and columns, and finally I take the square root. So we want to minimize the difference between Z times W and X. And this optimization is, of course, under the Z and W being non-negative. So we need this. It's a constraint optimization problem. And we do not have a closed form solution just as the eigenvalue decomposition of X transpose X for PCA. We don't have that, but we need some iterative algorithm to do the iteration. So for example, we can initialize some non-negative Z, try to solve for W using least squares, and then fix the W, we try to solve for Z, and then back and forth. And we have to be aware of that local minimum issue, just as we have for the k-means. So sometimes you need to try multiple initial values. This is the NMF. So the nice thing about this non-negative constraint is that mathematically we can prove that the solution of Z and W will be highly sparse. Okay, so the W is the loading matrix we're interested in. It still has the same format, that is each row corresponds to one new dimension, and it shows how it's combined from how it's formed by original P dimensions. So with a sparsity, you only have a few non-zero entries in each row. So then you can better say, okay, these original genes or features are contributing to my new dimension one. So you have better interpretability. Okay, so in this way, phi 1 to phi k. So I can say this is W transpose. Okay, has sparse columns. And this implies good interpretability. And some people actually use NMF as a clustering algorithm. Because if you say that phi 1 contains only a small group of genes with non-zero entries, phi 2 has. So then you can say each one corresponds to one cluster of genes. So we are doing clustering based on dimension reduction. You can see that in some bioinformatics papers. Okay, and recently my students found that there is another algorithm which is kind of in the middle between NMF and PCA, and that is called projection NMF, Pnmf. What's the motivation for doing that? So if we just do the regular NMF, you see that unlike PCA, for PCA, the only matrix we need to find is W. Because with W, we can just do X times W transpose, we get Z. So we don't need to compute Z specifically. We just need a loading matrix. But in NMF, we need to find both Z and W, and with the non-negative constraint, right? So it's the solution will be a Z and be a W. But this W is not necessarily a projection matrix. Why is that? So if we think of this, okay, so from NMF, the question is, can W transpose, which is K by P, be, P by K be a projection matrix Okay, that's the question. Can we do the same thing as for PCA? Actually, the answer is no. The reason is that imagine if you can let W transpose be a projection matrix, then we can say, okay, given that, then the Z would be the embedding or the projection result after we project Z of X to the K dimensions. That will be our Z. So in this case, what we would have is that if in this case we can write Z equals to X W transpose, then the objective, okay, this objective function or loss function to be minimized would become X minus X times transpose, that's the Z, times X. It becomes this. So basically, we're changing the objective if we ask W transpose to be a projection matrix. And this is not the loss function of NMF, but it is the loss function of Pnmf. So Pnmf is exactly trying to solve this problem. So it reduces the objection or optimization perspective by finding two matrices Z and W to finding only one matrix W. Okay, so here the constraint is just that we want to minimize X times X W transpose W in F norm, subject to that W itself is a non-negative matrix. That's Pnmf. So with the Pnmf formulation, we can actually use W transpose as a projection matrix. Okay, so then with new data, X star, we could do X star times W transpose with a Pnmf W, but we can actually not do it with the NMF W. So I think the Pnmf is a very nice thing. It has the advantages of both PCA by allowing you to do projection, and meanwhile, it has that non-negativity constraint so that W is very sparse. So that makes the interpretation of each loading vector much easier. And recently, my student has applied this to single cell data. So we just had a bioRxiv preprint posted. And so if you're interested, you can check out our result there. We can see that we can find very interesting interpretation for this new dimension. For example, some dimensions can be highly correlated with cell library size, and some dimensions can be super useful for distinguishing cell types because we found that it contains some highly informative cell type marker genes. Okay, so PC and NF and PNMF are closely related based on what we said. And if you're interested in that 1999, the original Nature paper, you can check it out. It does not have a biology application, but it is an image application, which is very interesting. So in that application, maybe I can talk about it a little bit. Okay, so let me say a little about the 1999 Nature paper. So what that does is that in their original matrix X, so the reason I want to explain a little bit is because the figure in that paper is somewhat difficult to understand. So if you don't understand it, if you don't understand the foundation here, then you will be confused by that figure representation, even though it looks very pretty. So in that case, the matrix M by P is corresponding to these are images, N images, and P are P pixels. So let's say that if each image is like this, okay, if it's like 200 by 200 image, each image I, okay, if it's a 200 by 200 pixel, then the P would be equal to I would just say 200 squared, okay? So that's a 40,000. So that would be the P. So what you do is that you just take the intensity of each pixel and do them in whatever order you prefer and concatenate this rows into a long vector, that's the vector in X. Okay, and then once you obtain the W, as we know, it's a K by P matrix, right? So, or probably it's, yeah, I'll just say this. So still, so here now I have K dimensions, okay? And the rows are still P pixels. Then what does it mean? So each new dimension will be a combination of the original P pixels. So to understand that loading, you can actually have each, I'm calling this loading K. For each loading K go from 1 to big K, you can plot it also as an image. So still a 1200 by 200 pixels based on the order, you stack them or concatenate them into a long vector, you just restore the order. And then for the entries here, the loadings, you plot them here. So for example, for this loading K, if you show, okay, the non-zero loadings, actually, if I look at the image, the corresponding image, it's corresponding to eyebrows of a person, then I can have a very direct interpretation to say, okay, loading K corresponds to eyebrows, and another loading vector corresponds to mouth, another loading corresponds to nose. So that Nature paper contains those very interesting illustrations. But you must understand that the illustration is done by restoring each loading vector back to the format of a 200 pixel by 200 pixel image. So you must understand that aspect to see what that illustration actually is. So I would say if you check the figure in that paper, that illustration is not a mathematical matrix notation, but it's just for this type of simple illustration. Yeah, so in other words, each image, each row in the original data matrix was a 200 by 200 pixel image. We put it as a vector. Once we obtain the loading vector to interpret it, we just stack it back into an image format. So we can see which pixels are giving high loadings in that vector. Then we can interpret the new loading or new dimension. Does that make sense to you? Okay, great. So that's all I want to say about the NMF.
Dr. Lee?
Yeah.
Can I ask a question about NMF actually? Sorry.
Okay, please go ahead.
If I'm understanding this correctly, is one potential downside of it that it cannot capture, say, negative relationships within a given, kind of dimension?
That's a very good point. Yeah, I see what you mean. You mean that, yeah, because in the loading, we do not have negative coefficients, right? We don't have negative loadings. So if you have, let's say, two genes that are negatively correlated, that is when one gene is highly expressed, the other gene is lowly expressed, right? I do not think they are going to appear in the same dimension, right?
Okay.
Because their effects are actually counteracting each other. But I think that can be considered as a nice thing as well, because basically in this loading matrix of NMF, you are encouraging that in each new dimension, you only include the positively correlated things.
Yeah.
So that makes the interpretation much easier, I would say.
Okay.
Yeah.
Awesome, thank you. And then I guess kind of a follow-up to that, are there, so I guess I'm familiar with things like probabilistic principal component analysis.
Yeah.
Does that give you confidence intervals on the loadings that, say, would help you interpret things where, like, if the confidence interval includes 0, then we say it doesn't significantly load onto that, whereas if it excludes 0, then that's kind of is that something that exists?
Yeah, I think that's exactly why people propose the probabilistic Pca.
Okay.
Yeah, and then you can use the likelihood as a way to interpret the result. So you can think of the Pca. So this Pca algorithm we introduced here, which does not rely on any probabilistic assumptions as an algorithm, just similar to least squares. And you can consider that probabilistic PCA just like what we assume as the normal linear model when you assume the random errors to be normally distributed with constant variance mean zero. So these are just two ways to look at the same thing. But when you look at the probabilistic way with the assumption that, for example, in probabilistic PCA, the typical assumption is that your data are multivariate Gaussian, right? Normally distributed from a p-dimensional normal distribution. So if you have that, yes, then you can assign confidence intervals with that assumption. Yeah. But the PCA algorithm, this algorithm itself is not depending on any probabilistic assumptions. So therefore, it's just a deterministic algorithm. And you can see that with that probabilistic PCA under the Gaussian assumption, the MLE for that loading matrix is just the loading matrix we computed here. Yeah, so that's another way to look at the same thing.
Awesome, thank you.
Yeah, thank you. Yeah, and also I know there are people who talk about the Glm Pca. So in Glm Pca, what you did is that you drop the normal assumption in probabilistic Pca, but you can assume your data follow some Poisson distribution like that. People have applied those to single cell data. Actually, there is a benchmark paper on genome biology about the different many dimension reduction methods and how they perform on single cell data. And very interestingly, people found that a lot of those newly developed methods didn't beat PCA, just the classic PCA. Yeah. Okay, so that's NMF. So the next thing I'm going to introduce is what we call multidimensional scaling, short as MDS. This is a very useful and classical method. I would say this is as important as PCA, but many people didn't know or learn this as much. So what's different from multidimensional scaling is that your input is not a data matrix, but it is a distance matrix. It's an n by n distance matrix. So we can actually write it out as D, okay? And we call its entry as Dij, okay? And I go from 1 to n, j go from 1 to m. So that's the distance matrix. So which means that when you do the embedding of n data points, you may not have access to its original feature vector, that is x1 to xn. You don't have that. You lose the raw data, but you have the distance matrix to start with. So the goal is to find some embedding in a low dimension, so y1 to yn. Usually, we're only interested in 2 dimension or 3 dimensions. So that means we want to find some easy to plot data points so that in the low dimension, the Euclidean distance between yi and yj is approximately just dij. That's the goal. So you want to preserve the pairwise distance with some low dimensional embeddings. Okay, so that's the goal. And in the classical MDS, what we do is by directly go from D to Y1 to YM without any preference on like the weights on these n data points. We just treat them equally. Let's take a look at the algorithm. And I can say that it is equivalent to PCA. Okay, so this is equivalent to PCA if Dij is defined as the Euclidean distance between xi and xj in our data. Okay, so xi, xj are p-dimensional vectors. So if we have that original data matrix, so that means this x, right? So we have xi transpose, xj transpose, and so on. If I have that matrix to start with, and we first do the computation of pairwise Euclidean distances between rows, as our Dij. And start from the Dij, we do the MDS. Then the embedding, we have Y1 to Ym, would be the PC, the first 2 PCs or first 3 PCs from PCA. That they are equivalent. But as I said, MDS is more general in that you don't need to compute Dij as the Euclidean distance. You could compute Dij as, for example, 1 minus the let me see. You can have it as 1 minus the correlation, right? Depending on whether you want to keep the sign or not, you could define as 1 minus the absolute Pearson correlation. So it means when the Pearson correlation is -1 or +1, you have 0 distance. Otherwise, you have a between 0 and 1 distance. So you could have other possible distances to start with from your raw data, or you may not even have the raw data. People just give you a distance matrix, for example, for some privacy concern. So in that case, you start with D, and then how would you find Y1 to Ym? That's the problem we want to solve exactly. Okay, so the idea is very interesting to know. Okay. So the idea is to find, the first step is try to find what we call a Glm matrix. So it has a definition. A Glm matrix of Y1 to Ym is defined as B. Okay, this will be an n by n matrix. And we write this entry as Bij. And what do we mean by a Glm matrix? It's just from the inner products of Y1 to Ym. So where this Bij is defined as Yi transpose Yj. So for every 2 data points as 2 vectors, we take their inner product and we define as Bij. And we want to call this the Glm matrix. So the idea is that if we can find a way to convert the D n by n distance matrix to the B Glm matrix, then we can just decompose the B to find Y1 to Ym. So we just take this route because it's easier to find the relationship between B and D. Let's take a look. So what we do here for classical MDS is that we write down the Dij, or sorry, we write down the, because we know that we hope this Dij to be approximate or at best equal to Yi minus Yj Euclidean distance, right? That's our goal. And then we look at, if we assume these are equal, then we take a look at what's the relationship between this Euclidean distance and B. Okay, so we have Yi minus Yj squared, because whenever we square it, it's easier. Okay, so what is that? If you think about it, let me write it down for you in terms of the dimension. So probably, I'll just say K, okay? So K go from 1 to, I'll just use 2 as an example. So let's say we'll have Y1 to Yn in 2 dimensions. So this will be equal to Yik minus Yjk difference square. This is how we define the square of the Euclidean distance. And we can actually show that we can open this square difference and write it as K from 1 to 2 Yik squared plus K from 1 to 2 Yjk squared, okay? And minus 2 times K from 1 to 2 Yik times Yjk. We can write this in this way. And this actually can be connected to inner products. We know the first term can be written as Yi transpose Yi. The second term is Yj transpose Yj. And the third term is minus 2 Yi transpose Yj, right? So we have this. And each of these can be connected to the entries in our Glm matrix. This is BII plus BJJ minus 2 times BIJ. Okay, so this is something very interesting to know. So if we want to set this to be equal, then we want Dij squared. This is what we have as input. We want Dij squared to be equal to BII plus BJJ minus 2 times BIJ. We have this. And we also have some nice properties about the relationship between about the Gram matrix, I should say. Okay, so moreover, for the Gram matrix, we have some interesting property. The first thing is we can consider its column sum. So for the jth column, let's say we take the summation of the n entries in the jth column. Let's see what that is. So we know by definition, Bij is just Yi transpose Yj, right? And we know that we can have this written as I from 1 to n, k from 1 to 2, the dimensions, Yik, Yjk. Okay, now what I can do is that I know the I is here, summation for I, and summation for k is here. A typical and useful trick is to swap those two. I first do the summation of I first, and then I do the summation for k. So if I do that, you see, I will get k go from 1 to 2, and I can put summation of I, Yik, okay, times Yjk. So I can create this. So one thing you know is an important property about Euclidean distance is that it is robust, or I should say invariant to shift. Okay, so this is invariant to shift, because you know that if I do the same shift for Yi and Yj, so if I do Yi minus a constant mu, and I do Yj minus a constant mu, you see that if I do the same thing, I don't change their distance. So their distance remains the same. So this suggests that I could actually standardize each column to have mean 0. And that will not change the Euclidean distance between Yi and Yj. So what I mean is that I could ask each Yi to have mean 0. That's what I mean. Sorry. I mean, I could for each dimension, probably I should say this. So what I mean is that, let's say that if the mu is a mu vector, okay, so if you think of this, so yi1, that's a vector, right? So yi1 minus mu1, yi2 minus mu2. If I do this, minus yj1 minus mu1, yj2 minus mu2. So what I mean is that I want to do the standard shifting in each dimension. So even if I do that, as long as the shift is the same in every dimension for both Yi and Yj, the two data points, I don't change their distance. So that means in each dimension, I could shift all the n data points to have mean 0. So I could do that. So therefore, I could set the summation I from 1 to n, Yik to be 0. Don't forget that I am going to find Y1 to Yn myself. So as long as these n data points, low dimensional, two-dimensional embeddings, keep the distance. I don't care whether they are centered at 0 or not. So that's why for simplicity, I just do this to make them center at 0. This is also easy for plotting. So with this, then you see I can have this as 0. That means the Gram matrix will have column sum as 0. Okay, so then based on this one, based on, let me see, I'm going to give it some mark. So based on this one, based on this relationship, what I can have for the third thing is that if I take the summation for dij squared, okay, so if I take the summation for I from 1 to n, then what I will have, you see, what I would do is I'm going to take the sum for bii. And the summation of I has nothing to do with here. It's just that this copy will appear for n times. So I'm going to have n times bjj. And finally, I'm going to have summation of bij. So based on this 2, I'm going to have a 0. And this is what? This summation of bii is the ith diagonal entry of the Glm matrix B. It's just equal to the trace of that Glm matrix B plus n times bjj. So this is something nice to see. I'm getting a simpler and simpler result. Then furthermore, what I could do is that I could actually have this thing. I'm taking summation for j as well. So double sum I from 1 to n, j from 1 to n, dij squared. So I'm going to have it here. So the trace b, because it has nothing to do with j, it will appear for n times, of n times trace b. And over here, summation j from 1 to n, it gave me trace plus n times trace b. So together it's 2n times trace b. Okay, so these results are all very important. So given that I have this result, I can actually solve for bij given the D matrix, the distance matrix. So basically, I can solve for bij. That is, I can have bij equals -1/2 dij squared minus d dot j squared. I'm going to define that minus di dot square. And finally, I think this should be plus d dot dot plus square. So what are each of these? So this d dot j square is equal to the average of dij square, where I let I go from 1 to m, but keep j, and I divide that by n. So I'm taking the average of dij square across I. And di dot square is 1 over n summation j from 1 to n, dij square. So here I keep I unchanged, but change j, take the average over j. And finally, d dot dot square is the average of all dij square. So I from 1 to n, j from 1 to n, dij square. So that's the overall average. Still, what I did is that I first take the square for all the distances, and I get a column average, row average, and the overall average. So if I do this operation, I can show that this is what I need for bij. So in other words, there is a one-to-one relationship between the distance matrix and the Glm matrix. So what I conclude is that is I can go from D to B, and my B is defined as Y times Y transpose. Okay, that's the definition of the Glm matrix right here. So yeah, so you see here the Glm matrix B and N equals Bij. So I should say that, okay, I should lay it out more clearly, because this Bij is equal to Yi transpose Yj. So then you can see if I stack the rows, of B. So that means just tell me, if I tell you I stack this B1J to BNJ, that's the jth column of B. I would actually have Y1 transpose to YN transpose times YJ, right? And that gives me Y transpose YJ. And then I further stack the columns. So you would have, say, B1 1 to BN1. That's the first column. To BN1, B1N to BNN. That's the last column, right? So if I do that, I would have Y transpose stays, and then I would have Y1 to YN. So therefore, that gives me Y transpose Y. So that's why this is the Glm matrix. So now you see, with this key results, I can go from D, or more specifically, the squared entries of D to B. And once I have B, what's left is to find what's Y and what's Y transpose. Let me see if I'm correct. Y1, oh, sorry, my bad. This is Y. Okay, this is Y times Y, yeah, because this is the n rows of Y times Yj. Right, and this is Y, and these things, I lay the rows of Y as columns. So yeah, this is wrong. It's Y times Y transpose. Yeah, that's the Glm matrix. And so what's left here in the second step of MDS is trying to find Y, okay, which is an n by 2 matrix or n by 3 matrix, depending on the dimensions we want, from B. How do we decompose B to find Y? And here we know the Glm matrix is a symmetric matrix, because if we take its transpose, it stays as B. So as a square matrix, we can do eigen decomposition. That's the thing we're going to do. So by eigen decomposition, we saw this for Pca. We have B equals to V times the lambda diagonal matrix times V transpose. Okay, these contains eigenvectors. The middle are eigenvalues. So then we can simply take the square root of the diagonal entries of this lambda. For each entry, we take its square root, and then we just do the left and right multiplication. So you can see if we set Y equals V times lambda 1/2. So that means for the eigenvalues, we take square root, still keep as a diagonal matrix. Then we can clearly see B equals Y times Y transpose. That concludes it. So the key idea is to go from the distance matrix to the Glm matrix, and then from Glm matrix to Y. And that is our embeddings. So Y contains n rows, 2 columns. Each row is 1 data point. And that data point has 2 dimensions. So we can just plot this Y to visualize those n data points. And that visualization will preserve the pairwise distances as provided in D. And we can see that when we have Pca, this is exactly what we are doing. Because in Pca, we also have that dimension reduction by eigen decomposition. So in Pca, what we're doing eigen decomposition for is for that X transpose X, right? X transpose X is what? It actually gives us something like, in the original dimension, it gave us the Glm matrix, but that's for the P features, not for the original data points. So when we do that, we're trying to find how can we preserve that Glm matrix of P by P features using a small number of new dimensions K. And once we find that, we use those new dimensions to encode our data. So when we go from P to K, we lose some information there. But in this classical MDS, we actually don't lose information. because every step go from here to here is exact, right? We have an exact relationship from D to B. And then we have an exact relationship from B to the eigen decomposition. Yeah, so that's it. As long as you have more, so this P of B, okay, so as long as in this lambda you have more than 2 non-zero eigenvalues, you're fine. So you can have this thing as n by 2 matrix. So you can try the MDS in R and just try the toy example in the R package, and you can see it's very interesting. And just as an extension, MDS has some variants. I'm not going to talk about those. In those variants, what you can do is that you can give weight to those distances you input. So you may want to preserve some of the small Dij better than large Dij, instead of treating all the distances as given or as accurate. So that can help you enlarge some distances in your visualization. Okay, so that's the MDS. Do we have any questions? And we can say this is a linear projection or linear visualization of distances. Okay, so before we finish today, the next thing I'm going to introduce is a very popular method nowadays. It's called tSNE. So that's for nonlinear visualization, and it is used a lot for single cell data. So before tSNE, we will introduce its ancestor, which is SNE. I think it stands for Stochastic Neighbor Embedding. And this SNE paper was by Vander Matten and Hinton in 2008. So it used to be proposed by visualizing some images, but later people use it for single cell data. And therefore, this paper has received a lot of citations because of that. So, oh, sorry, I should say the ancestors from 2002. Yeah, that's from 2002. And tSNE is from 2008. Okay, we should separate those two. So what does it do? Probably we should talk about SNE first before we talk about tSNE. So here we want to preserve not the pairwise distance as MDS does, but we want to preserve some kernel transformation of distance. By a kernel function, we're going to convert a distance into a probability density. So what we define here is this density as Pj given I. I and j are two data points. So what it doesn't mean is that it means in the high dimensions. So let me say the context. Okay, x1 to xn. The original data are high dimensional, just as in PCA. But we want to find y1 to yn in low dimensional, usually only two dimensional, just as in MDS for visualization. So we are going to define this density in the high dimensional space as the probability that xi picks xj as a neighbor given xi. So we use xi as the center, and we want to evaluate how likely is xi going to pick xj as a neighbor. So what we do here is that we use the normal density. So we hope this is proportional to, this is the density function of normal or Gaussian distribution of negative the Euclidean distance between xi and xj in the high dimensional space square. Okay, distance square divided by 2 times sigma I square. So sigma I square is some variance parameter we are going to define for point I. So in other words, we assume that this distance between I and j, or between I and any data points, follow a Gaussian distribution, and we just use this Gaussian density to convert a pairwise distance any point j from I in Euclidean distance in the p-dimensional space, convert that into a Gaussian density using this distribution. So this is the PDF of normal distribution with mean 0 and variance sigma I squared. That's what we are using. So you see, what does this conversion mean? If the 2 points are far away, when their distance is large, after the conversion, the density will be close to 0. If they are close to each other, let's say they are the 1 point, they are overlapping, distance being 0, then this density will give you 1. So you actually have that inverse relationship between distance and density. The smaller the distance, the larger the density. But this one itself is not a properly normalized probability, right? So you see, this is just a density, and it doesn't satisfy the probability that for xi, with that center, if it picks any other point in my data, then the sum of those probabilities should be equal to 1, because it has to pick one of the points as a neighbor. It's just that probability is proportional to this density. So therefore, for normalization purpose, what we do is that we just define the normalization as, so the numerator stays. So I'm going to keep it as this. And for the denominator, I am going to take the summation for all points that's not equal to I. Take the summation of the density, xk, to sigma I squared. Okay, that's the high dimensional version. And similarly, in the low dimension, I will define qj equals I in the same way. But in the low dimension, I just use the Gaussian density with variance 1. So in the low dimension, because y1 to in is my choice, I don't want to introduce the trouble of choosing what's the standard deviation again. So I just said that in the low dimension, I will use the density by setting the standard deviation to 1. So here, I would just use the distance of yi and yj in the low dimension. Okay, so this is 2 I will have here. And similarly, for k doesn't equal, oh, sorry. No, I think they actually remove, not 1. So variance 1/2, like that. So then this is gone. So I don't need to do the denominator in the exponential. So for k doesn't equal to I, then it's exponential yi minus yk squared. So this is the qji I want to have in low dimension. This is the pji I have in high dimension based on the data's Euclidean distance in high dimension. Now, of course, how to choose sigma I is a difficult problem to be addressed later. But the goal is to find q such that these two sets of distributions are close to each other. How many distributions do I have? For pj given I, for each I have a distribution. And that distribution is about which point xj would I pick as a neighbor. So I may pick any value, any point other than I. So therefore, this is a distribution across with n-1 possible picks. And each pick has a probability associated with it. So for each I have a n-1 value distribution, discrete distribution, and I have I go from 1 to n. So in the end, I have n distributions. So when I let I go from 1 to n, I have n discrete distributions in high dimension. And similarly, I also have n distributions in low dimension. Okay, and what I want in their objective is try to make these n distributions very similar to their corresponding n distribution in the high dimension. In other words, I changed the problem to make the two sets of distributions to be close to each other. And we know that in machine learning, a typical way of matching two distributions is to use the so-called KL divergence, short for Kullback-Leibler divergence. So what does that mean? It means that you have some reference distribution, and that reference distribution here is RP. That's our reference. And you want to find another distribution to be similar to this reference, and the distribution to be found is Q. So then it becomes an optimization problem to find the Q such that Q for each Qi's distribution is it's close to the Pi's distribution. And in that process, because Q can be determined by Yi and Yj. So therefore, we can change the optimization problem to finding Yi and Yj, or Y1 to Ym, such that these NQ distributions are close to these NP distributions. So in machine learning, almost every problem is ultimately written as a loss function, and that will be our loss function. So when we come back next time, we'll finish the SNE and we'll finish the t-SNE so that you will understand better what it's actually doing. All right, so that's all for today. Do we have any questions? Okay, so I'll stop recording right now.